unfolds. First then, without further ado, Anna and Annick, please come to the front. Anna Dumitriu works with the team of MRG Grammar, which is all about genetic research. And she has prepared a conversation with Annick Bureau, who is also part of a program disseminating a lot of the outcomes of the program. So please let us know about where you are at, Anna. Yes. I watch the time, yes. And can you do with one microphone, or should I give you the second?
We'll manage with
okay.
take two. Okay. So
please give them a hand.
So good afternoon. My name is Annick Bureau, and I'm the director of Leonardo Aulats, which is the French-European activities of the journal Leonardo. And Leonardo is one of the oldest journal publication dedicated to art, science and technology, and which is celebrating its 50th anniversary next year. And as such, we are very much interested about what's coming now, what's coming next, such as the FEAT project. And it's my great pleasure today to be in discussion with UK-based artist Anna Dumitriu. So Anna, you are known for your work with bacteria. She's a bacteria freak. And also for your work with and about infectious disease. You did a work in Vienna some years ago about plague, but your main disease, so to speak, is tuberculosis. And more recently, you have engaged in synthetic biology, and specifically with the engineered antibody necklace that you developed at UC Irvine, you created at UC Irvine. So before we discuss your FEAT project, could you maybe recall a little bit what you have done in the past and your approach?
Thanks, Annick. I just wanted to give a bit of background. So the whole thing I see as a kind of journey. I've got a long-term ongoing residency and collaboration with the Modernising Medical Microbiology project at Oxford University, which is in collaboration with Public Health England. And I've managed to get a kind of privileged access to microbiology labs as an artist. I trained in fine art, but I'm kind of, as she said, obsessed with the bacteriological world, particularly infectious diseases, because that's something that not a lot of people are really exploring, I think, or have the access to explore. So as I've developed this over the years, that's what I've become interested in. And this is an example here. This is my MRSA quilt, which is made using real MRSA bacteria and patterned with antibiotics in the process. So the bacteria, the MRSA, which is a superbug, is in interplay with different clinical and natural antibiotics in this. And it's telling the story of the scientists I've been working with, their microbial research. It's a kind of storytelling quilt in a way. So when people come and see the exhibition, or they can kind of have a discussion about what these different effects are, and because it's textiles, it kind of leads different audiences to it. As you mentioned, I'm quite obsessed with tuberculosis, and this is a piece which is using the extracted DNA of tuberculosis in its making. It's actually made of dust, and it's entitled "Where There's Dust, There's Danger," because in 1902, the advice of the British Society for the Prevention of Consumption, the old name for TB, was "Where there's dust, there's danger." They thought that dust was the primary cause. And I'm fascinated in the trajectory of how medicine and ideas in medicine change. And I worked with scientists who are implementing currently whole genome sequencing technology in UK labs and in the UK Health Service and beyond in order to track and study tuberculosis and as a diagnostic technique, which is going to revolutionise tuberculosis care. I did a residency at UC Irvine, as you mentioned, and this is the engineered antibody that's pictured. Each of the beads, I was trying to understand the synthetic biology work that they were doing, and they'd engineered an antibody to bind with HIV, but it contained 21 amino acids rather than 20 amino acids. So living things normally will only contain 20 amino acids. So this is something that would never occur in nature. They used the metaphor of beads and chains of these amino acids that then fold into proteins. So the DNA makes the proteins, makes the amino acids which fold into the proteins. And each of the beads in the necklace contains the actual amino acid in the sequence in a crystallized form and is in the exact order of the antibody sequence and then folded in the precise protein folding order. So you could, in theory, grind down the beads and reconstruct the engineered antibody. And I was really interested in the ways they were working with this. And most recently, I've been working with Professor Maggie Smith at the University of York in the UK, who is using something called phage integrases to edit antibiotic pathways in bacteria. And so this is another textile piece grown with streptomyces and patterned with viruses, patterned with bacteriophages. But also, I've worked with them to edit the antibiotic pathway in the bacterium so that it produced a blue antibiotic rather than a red one. And this is a kind of synthetic biology technique which will be incredibly useful for in the future when you'll be able to basically patch together different features from different bacteria to develop new antibiotics if this works as well as we're hoping. So that leads me on to, why I chose MRG Grammar.
Yeah, because as you can see, she moves through very serious medical and research projects. And for the FEAT project, you chose the MRG Grammar Consortium, which is targeting to decipher gene regulation. And so as you understood by what Eric and Lucas said, the project has started a few months ago, so we're still building up. So this already led you to Cambridge in the UK, to the Wellcome Sanger Institute, and you plan to go to Israel, to Haifa at the Technion University, and Tel Aviv at the Weizmann Institute. So could you explain what you expect, what you want to do, and why you are collaborating with those people?
Yeah, so I initially thought that the primary place I should go on the journey to kind of build onto my knowledge with what I've already been doing would be to go to work with the scientists at Technion, and with the EU projects there's always about 5 or more partners. And I was thinking to go there because they were the ones working with bacteria, and that seemed like the logical step. And then when I went to their meeting in Heidelberg at EMBL Lab, I met another scientist from Israel, but she was from the Siegel Lab, and this is a pioneering lab for microbiome research in bacteria. So the idea of doing the residency in Israel then split into 2 places, one where I'd be able to learn about using CRISPR technologies, this new gene editing technology that they're using a lot in the project, and then also at the Siegel Lab to see how they're working with the human microbiome research. And I was it's a strange thing, I was like a little bit wary of the work at Cambridge, because when I saw the presentations about it, they said, well, we're working with mice, and we're working to understand the immune system in mice. And I thought, how does that connect with my microbiology work, my bacteriological work? But when I thought, well, it's nearby, so I need to go there, and it's a world famous institution because that's the place where they mainly sequence the human genome. So it's a fantastic kind of place. But I went there, and it was absolutely the most wonderful experience because they were so open to my work and ideas, and it turned out that this is really the opposite side of the thing, because when you get an infection, you have an immune response, obviously. So I'm now studying how the immune response works, and they're trying to understand the different mechanisms behind how the genes in the immune response are regulated, because apparently, this is what they told me, that only 2% of your DNA codes for protein production actually does something that we understand at the moment. And the other 98% was what was termed for a long time junk DNA. So they didn't know what it was doing. But now they realise that it must be regulating that other 2% somehow. And there are genes that regulate the protein producing genes that could be 1 million base pairs away in the genome sequence. And the way that they regulate it is, you don't know where they are. They don't understand where they are in the sequence. It depends on how that protein, as I showed in this one, how the protein folds in on itself. And how the proteins fold is down to physics. So they're trying to use CRISPR again to knock out different possible sites of gene regulation to understand which one is the one regulating the gene. So they're creating mouse T cells that have different genes knocked out to understand which one's the gene regulation, the thing doing the gene regulation. And the scientist I was working with at Technion, he used a really interesting phrase of, if you can do it like this, if you can affect the gene regulation rather than doing these knockouts, you retain the integrity of the organism. So it doesn't have this thing. If you knock out a gene, something else might get switched on, something else might happen. But if you can suppress it in another way, it works differently. So yeah, it was an interesting term. With a different project that we're involved with, one of the artists was asked what was the thing that was most frightening her in science at the moment, and she actually highlighted gene regulation. Gina Janeski at the Trust Me I'm an Artist project highlighted this research into how genes are regulated as being the thing that concerned her most. So I was actually very interested to be able to kind of go in and research it.
So I would like to try to raise two questions and within the time that we have. The first one is art science collaboration, because obviously the FEAT project is about that, and the second thread is about your artwork and art practice and the aesthetic of it. And have you reflect upon it. So obviously art science collaboration and creation can take, and residency can take very different paths and different models, as obviously shown here in Ars Electronica and the different projects. And you have been also taken part in designing the FEAT residency, so I would like to have your reaction about that. But for the audience, I would like to stress upon one important thing for me, is that the FEAT, that is the Future Emerging Technology, the Scientific Consortia program, are over 4 years, started last year. And the FEAT project, that is the art in residency within the FEAT project, is over a year. So the time frame is rather different. So I would like to have you explain why you think this type of residency is interesting and why.
Through my past experience, I think the longer that you can collaborate with someone, the better. So I'm kind of obsessed with long, ongoing residencies. I've been working with the scientists I created the MRSA quilt with. We've been working together almost 15 years now. And over that time, you really can develop a language in the subject that you're working with. And so I think most of the collaborations I do, they tend to go on, and I'll come back to people, and they all end up collaborating in the end. It's quite strange because there are these cross links and they all get to meet. And so it was this idea of the long-term residency. Now that can't always work with professional artists. We're here, we're there, we have to travel a lot. And so I was at an event the other day and they were talking about, we do a residency program where artists get to hang around for 3 weeks in a farm with some facilities, and it's this lovely space for them to create. But I think most of the artists here will agree it doesn't really work like that, and that's quite a romantic view. So what's ideal about these is you can go in for a period of time, you can come out, you can work in your studio, you can develop stuff, you can work online as well, and then you can go back. And I think although there's this time difference, what I've found in all my other projects is that often the scientists will go and seek funding to bring me in further. I will seek funding to develop further projects. And so once the initial introduction is made and seed funded, which I think the funding for these isn't too bad as well, it's quite a fair amount. Once that's kind of seed funded, then the aim is that other things will take over and that the scientists will support. I've already had MRG Grammar people have already put me in touch with other EU projects they're working on and other funding streams, so to develop linked projects, which has been lovely.
Would you say that in an ideal world, with ideal funding, it would be better to have FEAT on the same time frame as FEAT, because the scientists obviously are going to develop other things and they're going into different directions than planned. So
in an ideal world, and I've talked about this before at the European Parliament, in an ideal setting, the artist will be brought in the development of the project before the grant application goes in, and will be part of the project throughout. And then would develop partnerships beyond that in future projects.
Yeah, I think so too. So in the time frame, we have 2 minutes. I would like to come now to your aesthetic and what you do. So obviously you are creating objects. And what is striking for me in your creation is that it's really craft. And it's often feminine craft, like embroidery, felting, things like, things like that. And it's mixing cutting-edge technology and science with very old techniques and DIY. And you once said that you were using everything you found in labs. So we'd like you to explain why you choose why you're doing this kind of output, so to speak, as an artwork.
I think really that each of the materials are really important to me. So I guess I trained in fine art, so I've been sort of trained under this truth to materials kind of paradigm in art, where you really want to bring in the reality of the material and use that. So I want to kind of tell the story of the work through the actual objects and materials. So I guess that the things like the bacteria or the amino acids, or in fact the background of this is dyed with Kumasi blue, which if you have done any gene editing you'll know is the dye that you use in electrophoresis to cut out different proteins and use that. Which originally it was a wool dye, and I love tracing these histories as well and learning how to work with them, and then trying to work out, are they safe to work with? I had a huge dialogue with some chemists about whether I should be just handling this myself, because they don't normally do that in the lab, and it has all these warning symbols. So I think the materials become their own kind of almost over-determined symbol within the work, I think. If you know what I mean.
Do you have time for one more question? So the last question. What also is interesting to me is that your work, your artworks embody, so to speak, very complex science. But at the same time, they are not standalone work. They need to be spoken about, and what they are as complex, so to speak, as the science they embody. And I know you're interested in communication and communicating science, and I know this is something you chose, that you have to talk about the artwork. So could you say a few words about that?
Yeah, I mean, for me, this idea of public engagement in science, I'm very interested that the work has that within it, which I know is quite a difficult and thorny thing, kind of is often debated. But for me, that element of it is almost a political thing. I think if people need to understand where these kind of new scientific disciplines or new scientific research is going, and also a lot of my work's to do with the past as well, where we came to this, and I think we often have to look back to see where we are now as well, to see where our ideas might be wrong and where we might be questioned, because 100 years ago they thought dust was the primary cause of tuberculosis, and I think the artwork needs to work on many different levels, I think, for me. And I like to retain all the complexity of the science within the work and then tell stories around it. And sometimes I'll provide text, but quite a lot of the time I run participatory workshops where members of the public will come in and sit with me and work with the scientists that I'm working with on the projects, and we collaborate to make things together. And this has a lovely effect of completely breaking down the hierarchy, because if you're at a sort of a science festival and you want to ask a question, you have to put your hand up in front of a big audience and ask a scientist a question. You're not going to ask those little random thoughts that you're having. But what we found is that it breaks down the hierarchy. The scientist is making the art, the members of the public are making the art as well, and they're chatting about the science, and I'm working with them to do that and the whole process. And that's something that I find very beautiful. When people come and they share stories that I would never expect to hear. Like when we did the MRSA quilt making workshops at the V&A Museum, we had a lady who was in her 80s come and participate, and she told the story of how she worked with Frontosil. I'm nearly there. Which was a very early antibiotic. So it was, and not how she worked with it, how she was treated with it as a child. It put me off. So that's what I like.
Thanks.
Thank you, Annick.
Thank you.
Yeah, sorry I have to interrupt you here. But thank you for this interesting insight in where you are. We're very curious about how we see the artwork in the end.